It's a real pleasure to have Pankaj here. A little bit more about the sort of official biography. He was an undergrad at Caltech, physics, and then did his
math major.
Thanks. And then PhD at Rutgers in condensed matter. And then, is that approximately right?
That's right.
And then Princeton postdoc with Ned, and wrote one of my favorite papers on signaling in dictyostelium with David Schwab, who, and now he's at BU. He's been there for 15 years. He's in the physics department, but he's, as I will tell you, very broad, and by virtue of being very broad, he's associated with many things. There's Faculty of Computing and Data Science, Microbiome Initiative, Bioinformatics, Regenerative Medicine. He's very broadly engaged with quantitative biology at BU and in the country. He's a fellow of the APS. He was a Simons. I'm doing the whole thing. Come on. He's like, we know each other. Okay. He's a Simons Foundation investigator in the mathematical modeling of living systems, Scilog Fellow, Sloan Fellow. He's a CZI investigator in theory and biology. He's really, for me, the intellectual leader in this field of sort of doing theory and biology. I just wanted to highlight some of his papers that I think are things that I remember reading and being really struck by, but more importantly, also their diversity. By the way, he was one of these people that was really asking, what is machine learning? How are machines learning? What is the physics of machine learning? Way before it became a hype. He wrote this beautiful review, which at least I have read tried to read. I use it as my reference. It's a reference in the course I teach, which is called High Bias, Low Variance Introduction to ML for Physicists. I love that review. You should take a look at it. Some other papers, Emergence, Simplicity in Microbial Community Assembly, completely different topic, microbial ecology, reinforcement learning in different phases of quantum control. Sounds like a different topic. This beautiful paper with David Schwab on exact mapping between the renormalization group and deep learning, which I think really got the field started in my view. And then energetic costs of cellular computation, and one that's very dear to my heart that he will sort of build on. He wrote the first papers where we try to build statistical models for cell states and cell transitions, and he continues to sort of pursue this in a very rich and beautiful way. He's a very fierce person. For me, Pankaj just is fierce. He's fiercely human.
Stop, I cannot live up to this introduction.
No, I think you're fiercely human, you're fiercely critical, you're very creative. It's a real pleasure to have you here. Yeah.
Thank you so much. I must say that it's hard to make me feel embarrassed, but you managed. Thank you for having me. I really appreciate it. It's been fun. Many of you don't know me. Those of you that know me should know that I really welcome. There's no reason to get through the slides. It's just ask me questions, tell me I'm wrong. It's meant to be a dialogue. I call this my scientific midlife crisis talk because that's what it is. It's my scientific midlife crisis talk. And so it's going to start with some kind of very broad philosophical stuff, and it's going to end with lots of data, like how you plot data properly. So if you're finding the beginning kind of annoying, I promise you there's some nitty gritty stuff about plotting data at the end. And I think that's, so yeah. So as I said, it's my scientific midlife crisis talk. And so, there's some intro, and then there's basically, I think what has been my implicit organizing way of these problems look very different from each other. So I think what we seldom do in science is give people access to the internal dialogue of what's philosophically motivating the approaches we take and what ties everything together. So that's going to be the second part of this talk. Then I'll spend very little time on this example for microbial ecology and spend a lot of time on thinking about development and cell fate specification, because I know there's a lot of people who are thinking about this implicitly. So, I think I'm going to start with this very usual thing, which is that I think one of the things that strikes any physicist that comes to biology is that it's really looks really complicated. And it's even more complicated than you think, because every time you zoom in on one of these diagrams, it looks like that. And this might have been the diagram Todd was showing me. No, it's not. That's not the ERK pathway. It's some metabolic pathway, but it doesn't matter. This is what biology looks like. And to a physicist, this looks astoundingly difficult to think about. And, and so I think what's amazing is, of course, that these are just some random pictures you're not supposed to understand, except to show you that people are measuring lots and lots of stuff. Because the idea is that if it's very, very complicated, we should measure everything and then we'll somehow tame the complexity. But then we don't have very good ways of representing this complexity in what we measure. And so, here's a UMAP of some single sulfate, some spatial transcriptomics, some, spatial validation. These are networks within genes. Every layer you look at, people are making amazing measurements. And they're measuring many, many things at the same time. But I would say we're lacking a real... At least I don't know how to interpret many of these pictures when I see them. Right? And the reason I think is that, as a physicist, I would say that biology is not physics, right? It's really composed of, importantly, heterogeneous components. So in physics, or in fluids or something, we're often thinking of many copies of the same kind of thing. Here you have many different copies of many different kinds of things. It works out of equilibrium. As John Hopfield always likes to emphasize, it's a matter with function. And it's a historical product of evolution. So it seems like, most of what we did in classical theoretical physics, it's hard to understand how to transfer all those ideas over to this biological setting. And in particular, I would say that the pillars of 20th century physics were really three things. There were symmetry, they were basically dealing with forces, and they were basically dealing with optimization and energy functionals. And except for, there are settings where each of these things applies, but on a whole, much of what I showed you on the last slide, it's hard to interpret through the lenses of these three major tools that we had in theoretical physics. And so the question is, how do you proceed? Right? And that's kind of been my, it's been my, I guess, my motivating thing. And the question is, how do we do theory? I haven't opened up a lab. And do we even need theory? So I about 10 years ago, I was reading this philosopher of science, Ernest Mayr, for the fifth time. And he just insists that you can't do theory for biology. It's not possible. You shouldn't even try. And that it's a foolhardy thing. And I think implicitly, this is the attitude of most of the field. That what we just need are better measurements, better statistics, and better computational tools, and the understanding will automatically follow. Right? And so, this is the age we live in. We don't have an oracle, but we do have ChatGPT, right? So I asked our modern day oracle, does biology need theory? And the answer was, yes, biology needs theory to explain the complex phenomena of living organisms. It's a very ChatGPT answer. Theories provide a framework for organizing and interpreting data. They help researchers to generate testable hypotheses and make predictions about future observations. Okay. Yeah, so apparently theory can do anything. There's a punchline to this slide. If I could measure everything about the system, would I know how it works? Right? Because I think that's kind of the ambition at the moment. We'll just measure everything, we'll know how it works. And it says, if you were able to measure everything about a system, you would have a comprehensive understanding of its current state, but you may not necessarily know exactly how it works. I don't know what that means. While careful measurements is important in understanding a system, there may be underlying processes or mechanisms that are not immediately observable or measurable. Basically saying, maybe you don't measure everything. And initially, even with a complete set of measurements, it may be difficult to discern causality or the relationships between different components of the system. I don't know what all this thing means, but the reason I read you this whole thing is that we know every single thing about at least some version of this program. We know every architecture, we know every piece of training data. We know every gradient prop, and no one can tell you how it does this. How can it generate such a coherent answer? Right? And so this is my retort to the people who say we just need to measure more. Here's a thing where we know every single thing about it, and yet we still can't tell you how it works. And I think biology is much closer to this than it is to anything else. Right? And so that's kind of, my ChatGPT AI parable, right? That here's something that can give us a coherent answer, but we know everything about it, and we still can't do it. And I would claim that it's unclear that biology doesn't live in this realm. So I think we need different approaches that aren't just machine learning, because building bigger statistical models isn't at least going to match what I want as my level of understanding. So how do we deal with this biological complexity? So what we've been taking, what we've been really drawing inspiration is from a kind of like a tangential branch of statistical physics. And it's something that was, pioneered by Wigner. This is a quiet audience, man. This is too much Midwestern sense of decorum. You should be like rowdier or something. So Wigner, right, had this kind of crazy idea. So he was trying to study uranium in the 1950s and 60s, right? And uranium's a really complicated molecule. And it's a very complicated nucleus. It has 92 protons, under 146 neutrons, and it's super complicated. And his basic idea was, okay, I cannot solve the Schrodinger equation for something this complicated. So instead, what I'm going to do is I'm just going to take this Hamiltonian that describes uranium and replace it by some random symmetric matrix. Okay, so it sounds like it's a crazy proposition, but it's a conceptually different thing. Instead of saying the state is random, as in normal statistical mechanics, we're saying the system itself is random. Now, what's interesting about this is that using this, right, he was basically able to explain something about real world data, right? So this is like nuclear resonance data from uranium nuclei. So these are basically scattering cross section. And every time, you see this peak, it corresponds to a resonance of the uranium atom. And of course, if you look at the distance between these kind of peaks, it agrees perfectly with basically replacing this complicated uranium with a random matrix that's symmetric. And the basic idea was that it seemed like, and this is now known as the Wigner surmise, blah, blah, blah. I think many people in this room are probably familiar with this story. But I think if you want to abstract away what he did, he said that complex systems are random systems, but in addition, you have to think about the constraints that are imposed upon them. So in this particular case, the constraint is unitarity. It's conserving probability. So that's why you have to look at symmetric matrices as opposed to non-symmetric matrices. And so the basic gambit that the group has been doing, I'll skip this part. This is basically saying that in the thermodynamic limit, things become universal, that this kind of replacing complexity by randomness is only really valid for very big systems. It doesn't really work if you have 5 or 6 things. But the basic gambit of the group for the last decade is that we should be able to think about complex biological systems should also be well described by appropriately chosen random systems. And the important point is that we have to really think about biological constraints. And these constraints can come from evolution, they can come from function, they can come from whatever you want. But the real thing is that it's not just randomness, it's randomness plus constraints. So and I'll show you in practice for the rest of the talk how we've been trying to think about this way of thinking about it. And what's nice about Wigner's thing, unlike the rest of StatMech, is that it doesn't require things like ergodicity or anything else. It really is just saying complicated things are complex, and then just think about the constraints that you must still satisfy. And if you do that, then you should be able to at least predict some properties of high-dimensional complex systems, and in particular, the generic properties. Of course, you can't predict everything about them, but there's some generic things that you should be able to predict. Thank you for the question. I appreciate it.
So what happens when the number of constraints get as big as the system?
Oh, yeah. So that's the question, right? The question is how many constraints you have to put in. So the way I view it, roughly speaking, is that there's some phenomenon that you want to explain in some data. And the question is how constrained you have to make the ensemble in order to reproduce the phenomenon. And what you'll find in these kind of very bulk measurements that you see is it turns out to be not that many. And you can either view that as saying we don't have good high dimensional null models for what we should expect in these kind of large scale measurements, or some other way, or saying that like it has to be generically true because these systems are evolved. And we're going to see examples of both of those in the 2 examples. The microbes are in the first thing and the other one's in the second one. Yeah, the question from
It's looking normal.
okay. Good.
Yeah, I think you're giving a clear talk so far, but I can tell you want some questions. So
Yeah.
it seems to me like would you endorse maximum entropy models? Is that kind of where you're going to go, or how is this going to relate to maximum entropy models?
I will not endorse maximum entropy. I am an anti-maximum entropy guy because maximum entropy is an optimization principle. And part of what I like about this approach is that it doesn't actually require any optimization principle underneath anything.
Although, In contrast, though, you would say that they're as random as possible, given some constraints.
Yes, I agree that it sounds very formal, but it's not even invariant to a change in bases, right? That's the funny thing about maxent. Entropy is not actually an invariant under basis change. So it sounds very principled. I would say in practice, it's not as principled as one would think it is naively. But no, I think part of what I like about this is that you don't have to assume any optimization, right? Wigner doesn't have to assume anything's optimized. But yes, in principle, you could
You
fit.
still have to specify the randomness that's present.
Oh, I agree with you.
Constraints, right? So
I agree with you. So I'll show you how we're trying to do it. Because we're not often thinking about probability distributions, because maximum entropy is really a different philosophy. It's really like I look at the states of the system and I assume those are random. And Wigner is really, I assume each system is random. And this is something, Wigner has a very beautiful review from 1967 I'm in an applied math department in SIAM. How do you guys say these journals? SIAM journals. There's a beautiful review from 1967 from Wigner where he quotes Freeman Dyson talking about this, and it's a beautiful review. I think it's in that I cited it in that philosophy piece that I just sent to Madhav. But and I think that's the distinction. So let me show you how it works in practice. And so this is going to be brief. And the first thing is just a series of works that we've been doing. It started off with my former postdoc, Charles, who now is running a $500 million company in Silicon Valley. And recently with my former postdoc, Bobby, who's now training to be a priest at the Vatican. And the group has been interesting. And Wenbing, who is currently a KITP fellow and back at KITP fellow and is going to move to Princeton in a month. And as another postdoc, and my friend Josh, who is running another company, and Alvaro Sanchez, who is now back in Spain running stuff. And these are the people who formulated all this. Yes.
So I was inspired by your vehement rejection of max entropy. So I want to take another step.
Yeah.
Is this going to be similar to, for example, like how one would model spin glass, or is it also going to be very?
Yes, it's closer to that. It's closer to that, but we don't need free energy functions. That's what's fun about it. So on a technical level, on applied math level, it's fun because you do all the disordered systems physics. But you don't need free energy functions. You don't need an optimization thing. You can do all the cavities and replica. Well, we don't even do replicas. We do cavities because replicas force you to have some sense of free energy in all this. All right. So this is old stuff. So I'll go through it quickly, except to advertise that I spent a lot of time writing this review on community ecology. For those of you who want to understand it, these are lectures I presented at Les Houches. This were written with Wenping and Bobby. I think it's very hard to find all this material in one place. So if you want to learn 1960s community ecology to now, and you want it pretty much spoon-fed to you, at least we tried very hard to do it. So and it starts with very low dimensional stuff and then gets to this very high dimensional kind of settings with cavities and disordered systems and intuitions. And one of the points of the lectures is to show how low dimensional intuition often doesn't translate to high dimensions and what translates and what doesn't. So these are, I don't know, I spent a lot of time on these lectures, so I want people to read them. We spend a lot of time on these lectures, so we want people to read them, and they're meant to be extremely pedagogical, I would say. They assume nothing. All right, so let me say, we've had these series of work, which you probably heard a lot about by now, because, so many people in Chicago are doing all these things where the basic idea was this observation that microbial ecology is shaped by cellular metabolism. And one of the things we wanted to say is that, metabolism is about consuming small molecules and then producing small molecules, right? And somehow that wasn't so appreciated, but the important point is that you take some energy in, use some of it to grow, and you spit the rest out. And this process is, of course, extremely, extremely complicated. That's the Keck database metabolism thing I showed you. And a la Wigner, the basic idea of all this stuff was going to be to take, just like Wigner said, this is very complicated. Let's replace it by a random symmetric matrix. What we're going to do is we're going to say physiology is very complicated and metabolism is very complicated. And we're going to replace them by random matrices. So we're going to replace consumption with some random matrix that tells you about how much I consume each thing. I'm gonna take metabolism, and I'm gonna say that's some random Dirichlet matrix that tells me how I partition energy into stuff, and then I'm done. All right. Of course, I haven't told you anything because I haven't told you what ensemble I'm drawing from or what dynamics are going on, but we're gonna get there as we get along. But the basic idea is, it's all complicated. Let's replace it by a random matrix. What can we tell you about real life or anything, right? And so the basic kind of equations we're thinking about are equations that kind of look something like this, where you have species I whose abundances that grow proportional to how much resource they eat. Right, so it's just like I eat all these resources, R alpha, with some consumption coefficient. I grow here. I take some fraction of the energy and I leak it back out using this random matrix that encodes as metabolism. And then this is basically the whole dynamics, right? And now what I'm going to do is I'm going to let there be many species and many resources. And now I'm going to play games. Like I'm going to ask, how much structure do I have to put into these random matrices in order to reproduce these patterns I see in large scale sequencing? That's basically the only game in this whole thing. Yeah.
Maybe Stu, but is it clear that it has to be linear in the resources?
No, this is just the simplest.
Right. Okay. So that's the first assumption you made.
That's the first assumption. There's many assumptions I make throughout, yes, but it's mostly because we are saying that most fluxes, and yes, we're ignoring saturation, and what that gets us is that these things are often not very chaotic or multi-stable. But yes, in principle, you could put much more complicated things. The idea is that around some reference resource, I shouldn't have to worry too much about it. But yeah, I mean, that's the least of the ridiculousness in this model. Yeah.
Just to be clear on the Wigner statement, there's the randomness and then the constraints.
Yeah.
The dynamical equations also act as constraints.
Yeah, these are the constraints. So
Those are the, but then you also have structure that you will put in.
Yeah, exactly.
So there are some constraints that you will bring about there too.
Yeah. So the main constraints I'm
Are these
arguing
equations?
are basically flux balance constraints.
Right.
So what these things are basically saying is that fluxes of energy in the ecosystem have to balance. Conservation laws. They're mass and energy conservation. Right. And then other constraints of evolution, I don't really know how to deal with. So we deal with by putting in structure into these random matrices.
And in terms of, are you keeping the ratio of N and R fixed, or you're letting both of them be large, but
Well, yeah.
both are just large?
Both are large. The ratio is fixed. If you want, I have no analytics, even though we solved this whole system with cavity. It's the best calculation I've done in a decade. It was so good, I didn't even send it to a journal. I just put it on the archive, but because I didn't want to ruin it with the refereeing process. I love that calculation too much. And Bobby's training to be a priest, so he doesn't need the paper. So it just sits on the archive. But yeah, I mean, you can do technical calculations. I'm just going to show you simulations because I think this, especially for this part, it's going to be it's more philosophical. I'll show you more calculations and data analysis in the second part, right? There's tricks to even simulating this in high dimensions, I want to add. Yeah, so the point is like the kind of things you can imagine putting in is that, oh, there's no structure in these things, or you can imagine that there's, different tiers of things like families that are specialized for different kinds of resources. So for example, sugars, carboxylic acids. And in this kind of setting that just corresponds to how much block diagonal structure or different things like that you have in these things. So you can imagine, you can tell yourself stories about what kind of ensemble you want to draw these things from. And the nice thing about all this is that you don't have to tell the stories yourself. We spent a lot of time writing a Python package that makes this extremely easy to do and simulate in high dimensions. And it uses a lot of tricks where we figured out that you could map all these problems. Instead of forward integrating the ODEs, you can actually map them to a series of optimizations using something like expectation maximization that allows you to actually simulate at scale very easily and deal with the fact that generically these ODEs are extremely stiff. And so there's some duality with constrained optimization that we worked out that actually makes this run fast, and which is why we can use this to actually test hypotheses very easily. Because if you had to forward integrate the ODEs, it would never work. They would basically all collapse on you over and over again. Yeah.
You always look at fixed points, right?
here in this stuff I'm going to talk about today, we've been spending more time thinking about chaotic dynamics and when you get chaotic dynamics and things like that. But that tends to be very far from the experiments. The stuff we've done on chaos has very little to do with explaining data. The stuff on explaining data tends to be all about fixed points. I would say, but we have a recent PRL, my undergrad put out on how you get chaos in these consumer resource models and a generic, you get a generic thing that okay, I can't even explain the bound to you. It would take me like 10 minutes, but then we can come up, it's a very nice conjecture. If there's some mathematicians in the room, I'm happy to explain them to you. All right, so we started by basically using this to design a bunch of experiments that Josh was doing. This was published in Science. I don't want you to understand everything, but it was one of these things where both Josh and Alvaro would never believe that the model could reproduce qualitative things. And it's a shame because we did the theory first and did the experiment second. But of course, the paper is written in a very inverted order to the order in which things were done. But it was just to show you that, like, you know, I can't tell you what species or what resources do, but the qualitative phenomenon seem to be very well captured by this random matrix kind of formulation that I showed you. And that was kind of very surprising to us that it worked so well. So then we wanted to go and ask, like, we thought this would just fail terribly. It would be like, oh, look, we motivated these experiments. We can't actually qualitatively reproduce stuff. So then me and Bobby spent a lot of time trying to think about, can we describe other large scale sequencing things? And so one example of the kind of thing that happened was like right around when we were playing with these models, this thing called the Earth Microbiome Project came out. And what it was one of these classic biology surveys where they went out and they surveyed lots and lots and lots of communities. And they just measured what taxa were prevalent, what species. It doesn't matter, really. The color you can ignore for our purposes is just the kind of biome it came from, Earth associated, whatever. And what they found was that that if you look at each of these columns is a different sample, it's a different environment. Each of these rows is taxa, and you can see that what they found is what they call this nested structure, which means that the taxa that are found in the most diverse community, in the least diverse community, seem to be a subset of the taxa that are found in the most diverse community. Just as if you find something over here in these kind of not very diverse communities, you're very likely to find it in these very diverse communities too, but not vice versa. There's many reasons this could be true. Sampling, it turns out that it's pretty convincing that it's not just sampling effects for various reasons. So we want to understand, can you get this? And the answer is, as you would probably expect, it's almost impossible not to get this. So if I run some simulations where I just go back to these equations, I have them somewhere here. And what I do is I do these kind of, the way these simulations are set up is I have some big pool of species, which I call this regional species pool. And I just take different environments and I seed them. I make them slightly differently. And I just take some random subset and see who survives. I can basically reproduce this idea of what's going on here. And you get this upper diagonal matrix. And in fact, what turns out to be true is that it's almost impossible not to get this, right? So we had a very, very smart referee who was like, yeah, you can produce the patterns. I don't care. Tell me how you break the patterns and then I'll learn something. And it was like one of the best referee reports I've ever gotten. It was a throwaway comment. So we spent a long time trying to break this pattern and we could never do it. Until one day, Bobby figured out there's exactly one way to break this pattern, which was that in these communities, the reason some species survive and other species don't survive is that if I put lots of species in here, they kind of compete with each other, and some things are fitter than other things, and so they exclude each other. But imagine instead, what limited the species that got into a community was not competition, but what immigrated there in the first place, dispersal. So as soon as that happens, you actually break this pattern. So it indicates that probably it gives you a direct ecological hypothesis, which is that it's probably that this pattern reflects the fact that ecological competition is much more dominant for shaping these things than dispersal. Right. So these are the kind of games I think I like playing with these random matrix theory things. It allows you to form good null models and formulate hypotheses. So here's another example from Otto's lab. Was Otto just here? Otto was here last week somewhere, right? In an ITMB or something. He was here at your guys' center, huh?
Couldn't make it sadly.
Oh, he couldn't make it. Okay.
He's in Vienna.
Okay. But this they had these nice models where they would take these kind of ocean communities and they would grow them on these agarose beads with a single carbon source. And what you see here, the columns are the taxa, the y-axis, the columns are time, the rows are taxa. And what you're supposed to see is that bright color means things survive. And so what you're supposed to see is that there's succession, meaning the things that come early don't come late. Now you can ask, if you read the paper, there's a lot of stories about chemistry hanging out here. And you can, please ignore the right side. I'm running out of time, so I'm gonna ignore it. But now you can ask what happens, how hard is that to get? Well, you just put random matrices in, it's impossible not to get that. As long as you start with a simple environment, right? So the important point of these things is they have a single carbon source, so they're a simple environment. If you have a simple environment, even without any structure, you're always gonna get succession. It's only when I have a really complicated environment that you don't get succession. So now you can say that's kind of obvious. Succession is about complexifying the environment, but this is a way to make this slightly more quantitative and test these things. And there's other versions of this. So if you go to the scientific reports, it's full of patterns drawn from everywhere. And we were kind of very surprised that we thought it would be very hard to produce these patterns. Our experience is that it's almost impossible not to produce them. You have to spend a lot of time thinking about how to break the patterns. And that in some sense is nice because it tells you, it gives you a way of generating these ecological hypotheses that are hard to generate otherwise, I would argue. So that's kind of the sense in what we're using typicality, and it's hard to put something like this into a maxent approach because it's really not about maxent at all, is what I would say.
Did you have to put a lot of structure in the matrices themselves, or these are really random?
These are really random.
No rank, blocky.
So you had to put structure in if you wanted this thing they called modularity, which was that if I grow a community on one,
Right.
and the other, that the mixture grown on the mixture looks like a random combination. So if I want that, then I have to put structure. But if I don't put structure in, I don't get that, which is fine. But what I wanted to point out is that these phenomena have almost nothing to do with the detailed chemistry. It's not the way most people would interpret these experiments. When most ecologists or biologists look at them, they tell you a story about chemicals.
I think there's a subtle thing though that you do in these papers, which is if it, this thing that, okay, it's not a molecular question about a part, but the existence of successional dynamics, and that is a thing to explain, and that a random null model can explain it. I guess I'm just saying that's still the art of this approach, which is what observable is the thing you decide to sort of describe using a sort of random approach. I think that's a little.
No, I agree. I mean, what I'm saying is that it tells you what you should be surprised by.
Right.
Right. I mean, which is, I think, another way. That's my more positive way of putting your negative, not negative, but my more positive way is like, often when I see these experiments, I'm not sure even should I be surprised or should I not be surprised? And what I like about this is it allows me to at least say how, on some level, make some small progress towards that. Yeah.
So this is a philosophical question, but I wonder if it's also a question about if we're asking figuring out the right ways to try to summarize data coming from these experiments, because people take a lot of complicated data and they want to try to simplify it. So they look for some low dimensional way to summarize what they're seeing. I wonder if there's some correlation there with you take a random system and you put some small number of constraints, and that's enough to give you the those low dimensional statistics that look similar.
Yeah, I think that's basically what we're saying, that like, in some sense, the number of constraints for the level of the data we're getting is not as high as you think. So, right? It's that because it's this sequencing high throughput data, though it seems quantitative, is rather crude in some level. It's really mostly survey data, if you really think about it, even at the level of gene expression or the level of microbe, that surveys on some level are insensitive to much of the detail, at least the qualitative phenomenon. It's really about high level constraints that you put on the ensemble. I would agree perfectly with your summary of what the way I like to think about these things. So this sometimes goes over very well with biologists, sometimes goes over really poorly. But I mean, but I think on some deep level it is kind of an interesting thing to think about, right? Like, what can we really learn from this data? So I'll show you the last. This ends at 15, right? I have until 15. All right, good. I have enough time.
But I started a few minutes.
So in 20 minutes. So this part, I'll show you a different thing. In the last one, the constraints were almost missing, and the randomness was everything. Now I'm going to show you a different thing where the randomness is almost missing, and it's all about just, I'm going to get the constraints right, and I'm going to define them to you. And then I'm going to show you that this one is really surprising. This all happened because my student Maria ignored all my protestations that nothing would ever work and just did things anyway, which is, it's a lesson for you grad students. You should ignore your advisors. Sometimes, exactly. The art of this is you should know when to ignore us and when not to. Right? It's like the art of the randomness. You have to know when to ignore the complexity and when to take it seriously. So this is a series of things that started way back in 2014. I stopped working on it because I was so disappointed by how this was perceived, and then actually got dragged back in by my friends Laertes and Daryl Cotton, because they came to us with a very practical problem. So they're lung biologists. They want to make lung cells they can transplant. And they were using all the standard tools out there. And they knew that the cells they were making in the directed differentiation protocols looked very different from real cells. But all the stuff that was out there was basically homogenizing them and telling them it was the same. They were like, come back, help us. And then also they took very seriously my idea of spurious attractors, and it was just very nice. They like, at least they took us very seriously, and they had this very practical problem. So we came back and started working on this again 3-4 years ago with single cell RNA-seq data. So I'm going to tell you about this thing, and because I give this talk to so many audiences, you will bear with me that I will give you 5 minutes of the very dumbest version of biology there is, which is that we're interested in different cell types that express distinct genes. And of course, the important point is that different organs, or different cell types are defined by different distinct gene expression profiles. This thing is, how this happens is very complicated. It depends on space, time, and all these various things that are going on. Most of what I'm going to tell you is going to focus mostly on this organ here, which is lung. And that's just because I don't have a particular love of lung. I just have a particular love of my collaborators who happen to work on lung. And, as I want to emphasize, I know many people in here work on single cell gene data, but I think the thing that just every time I work with this data that strikes me as so crazy is how high dimensional it is, and simultaneously just how noisy it is. It is so noisy. You just can't believe that 30% of something can be zero, but that's a pretty typical number, I would say. So it says you measure 20,000 genes, but at any given cell, 30 or 40% of them will be zeros. That's been our experience. And of course, what's been going on for a long time is that, one of the big projects over the last 5 years has been these compilations of human cell atlases that basically measure different cell types, and they measure all these genes and cells. And I think one of the things that is kind of hard to understand is how you take these atlases and how you get some way of taking this data and turning it into something about development. At least to me, it's very, very extremely difficult to go from atlases to development, even though they're measuring things in space and time. And so, all I want to say is that we have this idea of gene networks, and the inputs to these things vary in space and time, and somehow from that you get this kind of developmental process. And this picture in atlases, it's very hard to understand what they have to do with each other. And there's this gambit that Conrad Waddington made. I guess it was Crick's gambit, the way I'm talking about it. Madhav has made me self-conscious. I have to rewrite all my talks now. Because he pointed out that Waddington meant something very different from this, which is true. And I should have realized it because I read The Strategy of Genes like a year ago. But Crick's interpretation of Waddington is that you kind of have this gambit that Waddington was talking about, which is that he was saying that maybe we don't have to think about every kind of cell or every kind of gene interaction, but instead we can think of a more abstract thing, which you can think of as a developmental potential. And I would say that this is the emergence gambit. It's that I don't have to worry about every interaction as long as I get this kind of basic idea of development as a dynamical system right. I'll be able to understand everything without understanding all these details that lie underneath in the second picture that very few people show in Waddington's landscape. So the basic idea is that development is a dynamical system, and the attractors of this dynamical system in some sense correspond to different cell fates, or as Madhav pointed out, it actually corresponds to different embryos, which is 100% right. But in my interpretation, they're going to correspond to different cell fates. And again, the idea is that, cell types are a dynamical system, and that you can think of a phenotypic landscape that results from genes and signals. And what we should say is that we won't have to worry about all these genes and signals if we get the landscape right. So I promise you this is going to end with a lot of plots of data. So the thing about Waddington's picture that I just never understood, like it makes lots of sense, and I've reread this book at least 3 or 4 times, is like there's no coordinates and there's no scales. Right? You have no sense of distance. You have no sense of what's time, what space means. And I would say that actually the problem is that the way we plot this data now, I can ask the same question. What are the coordinates mean and what does the scale mean? So this is, this is 2 data sets I'm going to talk about. This is from the Lung Development Atlas. And I'm going to focus on basically this transition where you have a progenitor cell that goes to AT1 and AT2. This is a second data set, which is this Larry experiment from Alon Klein's lab where they barcode a bunch of homeopoietic cells, and then they basically try to figure out how progenitor homeopoietic lineages appear, right? But this is basically how the data is presented, right? In this kind of UMAP fashion, or this is a epithelial diffusion, this is UMAP. But it's very unclear what the scale means and what the coordinates mean. And it's much worse than that. By including this club and ciliated here, you make these coordinates look much closer than they would otherwise if I had just put them apart. So it's not only that the scale, it doesn't have scale and coordinates, it's that the scales and coordinates are often actively manipulated, I would say, in the way that data is presented and you can do that by including or not including data sets. All right. So what we've been trying to figure out is how do we properly mathematically formulate this and use this to plot data. So that's what I'm going to try to show you. And the basic idea is going to be this gambit that we're going to try to combine these atlases with this idea of development as a dynamical system. And coming back to this basic idea of Wigner, is the basic idea we're going to say is that instead of thinking, I don't know what the actual developmental network is, but I want a network that respects the constraints. And the constraints in this type are that I want a dynamical system whose attractor corresponds to the known cell types. Right? So it's all constraints. So I'm going to say any dynamical system whose constraints I understand, whose attractors I know are going to correspond to these things. And so there is a version of this that is well known in physics, which is called the Hopfield model. And the idea is that you model gene interactions as some random network with constraints that are attractors, and you choose the network to get the desired attractors, and the desired attractors are going to come from these atlases. So this original version of the Hopfield model doesn't really work for various technical reasons I'm happy to get into with whoever wants to get into, but we're going to update this basic idea with some kind of modern Hopfield networks. So there's certain challenges that you have to face on a technical level that are worth thinking about. One is that the data is very noisy. The second is that the attractors, the attractors you want to put in the different cell types are highly correlated. So that makes it very hard to use the classical constructions that people want to use. And so, so the goal is to model gene networks as a random network with constraints that the attractors are gene expressions from cell atlases, and we have some kind of very funny construction that actually does this, and it's kind of interesting. And the basic idea is that let xi note the expression of a gene, and then this zeta, what is this? C mu I is the expression of gene I in cell type mu, and this we get straight from the atlases. And then what we're going to do is we're going to make these things we call order parameters, which are basically magnetizations. They're basically dot products between mu I and xi. And then we're going to basically write a potential and some update thing, which is called a modern Hopfield network, and some dynamics that I'm going, that basically is a gradient dynamics in this magnetization space instead of in gene space. Right? And there's nothing tricky. I'll show you some examples of this with some data, but it's summarized in this kind of thing like this. So you have three spaces. You have a gene expression space. Using these profiles, you can go to a sulfate space. In this sulfate space, you have almost a gradient-like dynamics. So you have some potential. You take the gradient of it, but then you have to put it through a nonlinearity. And all the dynamics happens here, but then I can go back to gene expression space by just using my cell type coordinates. And the basic idea of what you're doing is you're basically saying, I take my gene expression and I break it into a basis of the sulfates that I care about, plus stuff that's perpendicular to that. And all the dynamics of sulfate decisions, we use the notation here, all the fate of sulfate decisions basically happens in this kind of sulfate decision kind of things. In this kind of space of sulfate. So it's a weird construction. You have to just trust me that it does what it does. We don't really understand why it works. It's a generalization of the stuff on modern Hopfield networks that Krotov is doing. And the important point is that this potential here actually can just be parameterized using normal forms of different bifurcations. Right, so this potential, of course, changes in time. That's what bifurcations are, but we're going to parameterize it due to these signalings. So it's almost gradient. It's almost gradient dynamics, but it's put through a nonlinearity. And the important point is to distinguish between these two spaces. That all the dynamics really is happening in the sulfate space, but we observe it in this gene expression space. And the sulfate space is a low dimensional plane in this full gene expression space. So how do we analyze these to use these ideas to analyze single cell RNA-seq data and assess cell similarity? So the basic idea is that we just construct these atlases, right? And the only trick that we do in all these things is a normalization trick that I'm not going to tell you about. The only trick we have is that we normalize each cell to itself. So instead of thinking about the absolute level of a gene expression, we just basically rank order all the genes in a given cell, and then we assign each gene a z-score. And so now you've given up any hope of knowing anything about a gene, and you're defining a cell by the ordering of what percentile is, say, OCT4. It's like, has a z-score of 3 in embryonic stem cells and -2 in everything else or something like that. And then we just basically construct these magnetizations. And the idea is that now we can basically go from this gene expression space, then you have a cell type subspace, and we're going to plot everything on this cell type subspace. And each cell type is basically just defined by the atlas. So how does this work? Right? So the idea is that once you have this thing in the cell type space, you can basically, make plots of how this gene each cell in the cell type space, you can use it to basically figure out scores, how similar this question I was telling you about that motor back, what are the scores of different things? And you can go through and ask what's happening. So here's like a very typical thing from epithelial lung cells. So here, what I've done is I've taken a bunch of single cell cells. And they correspond to these different cell types, AT1, AT2, basal, ciliated, it doesn't really matter. You should know that they're pretty closely related. AT1 and AT2 are almost the same cell type. And what I've done is I've just calculated these projections. And you can see that, if this was noiseless, or if we make pseudo bulk, this ends up over here at 0.8, but because single cell things are very noisy, you can see that these cells that have been labeled using marker genes look like AT1 sit here over here. These AT2 genes sit over here. The cells look over here, and you seem to be able to resolve these things in the cell type space. And now if I change the axes, because I can project on all cells at the same time, here, now you see that the AT2 goes away, and the ciliated cells project strongly as ciliated, but they don't project on anything else. So now the axes have scales. One means something. They have meaning. And all I've done, again, is just go from gene expression things to cell type space. And so the kind of fun things that I like is that, so this is something from the lung atlas, and they basically watched lung development. So they basically, sequence these lung things at different time points, embryonic day 12 to 14 post-birth. Yeah. I have 5 minutes. Yeah.
Okay to ask?
Yeah, go ahead.
I think this is very nice, this projection on final cell type, but maybe I could just, it's a quantitative question, but maybe it's a philosophical one as well. If I was thinking about the time series of development, where the terminal cell types are your space in which you get your magnetizations, do you initially think that early on in development, there are more dimensions that then condense, so to speak?
Yeah. Oh, yeah. That's going to be all the rest of the plots of the talk that you haven't seen because they weren't in the paper. Right? So this is basically, instead of diffusion maps or UMaps, this is how you can watch these embryonic stem cells. So the top is early epithelial progenitors, and it's a three-dimensional plot. I can plot this in whatever you want. And what you can see is you kind of start in this cloud, this thing bifurcates, and then you end up with just AT1s and AT2s, right? So it's this kind of crazy bifurcation. And what's interesting about it is that actually this axis has meaning. All of a sudden I can see movies moving in spaces that make perfect sense. And I'm going to show you lots of examples of this, right? And this is just a 2D projection onto the AT1 and AT2 space. And you can see that there's these funny transient states. So it looks like I might be seeing a bifurcation if I thought about it a little bit. So I start here. They seem to move out to the middle somewhere. And then they kind of go out that way. And then you have these cells that stick around for a while here. Then you get the air-liquid interface. And then they kind of disappear.
So since you're projecting into the defined cell states, do you learn anything about the intrinsic dimensionality besides what you impose from the projection? Okay.
Intrinsic, well, there's no reason.
What I mean by that.
There's no reason it has to work. I mean, what I would tell you is you do learn something intrinsically about the size of the bifurcation that's controlling the decision, which I have which I'll show you. You can believe me or not. I think what is surprising is you don't need any nonlinear anything. You just do linear algebra and non-orthogonal coordinates, and it seems like you can view everything. So let me show you some more plots. The rest of the thing is just plots. But before I do that, I just wanted to point out that our construction essentially allows us to put in different bifurcation types here, because there's a potential here, which is basically the normal form is defined by the normal form of the bifurcation. And so, you can think about some, bifurcation where you first turn on one signal, two things collapse, then you turn on another thing and you get something like this. And this is, what Rand and Sigia basically call this the triple cut. Is that in any other papers? It might not be the triple cusp. Then there's these heteroclinic flips, which do different things. And the basic idea is that this is one network, right? What I want to emphasize to you is we just came up with a single construction that has all the features we want. Whether this is a construction of biology, I don't know. But what's surprising is if you go and plot data, right? So this is the same data I showed you, the lung development data, and this is a simulation of a triple cusp bifurcation. And these are simulations where we just drop 30% set 30% things that are low expressed to 0. And what you can see is that this is the same data plotted from 3 different things. And it's shockingly, I'll show you more. You'll believe this more because I'll show you different bifurcation classes. All we've done is plot data. And you can kind of see, especially in these kind of things, it kind of looks unsurprisingly, somewhat surprisingly, as something that looks like a triple class bifurcation. We don't know what to make of it, but it is kind of shocking. You can do this. So let me show you another bifurcation class. So now you can go to this kind of barcoding data from a lone Klein. And one of the things they really found was that there were these kind of progenitor bivalent progenitors that seem to give rise to multiple kinds of cell type. And so you can so we have the barcodes, right? And the way it was usually visualized was something like this. You do a UMAP, these progenitor cells somewhere sit over here, you'll have the barcodes and they go somewhere else. But it tells you it's very hard to understand what the nature of the bifurcations or what's going on here. So here is a monocyte to neutrophil differentiation, right? And so I just want to emphasize the bottom are just plots of data. We've done nothing. It's just plotting data. And what you can see is that this
you're not being fair to yourself. You've plotted the data in a way informed by your ideas, which is not just UMAPs.
It's not just UMAPs, but what I want to point out is there's no statistical fitting. It's literally just plotting data in some basis functions defined by the atlases. And what you can see, and these are just different views again, and what you can see is that this thing actually, if you stare at enough of these things, it's single cell RNA-seq data. It's extremely noisy. I can't believe you can see anything, but it looks kind of like a heteroclitic flip. And if you go to a different thing, which is like a basophil to neutrophil things, you look at them and they look much more like a double cusp bifurcation. But the point is that you can kind of see these bifurcation types just by plotting the data. It's rather, there's signatures of it, like, and that's something to aim for, right? If we can really classify these cell fate decisions by different kinds of bifurcations, which is basically the philosophy of a whole set of people that I think that are very smart people, right? But a way of classifying cell fate decisions by the universality classes of bifurcations, then there should be a way to get it from data. And what's surprising is that you see the signatures just by plotting the data without doing any statistical fits. And the gambit, of course, is the reason this should work is that the gambit was that any network, any model that has the right constraints, if I can get a plot that's inspired by that, then I should be able to see it in data because biology is an involved system, right? It's kind of the gambit we make in physics. So that's the Landau free energy. It doesn't really matter. As long as we get the symmetries right, we're going to see it in data. And here we're saying basically, as long as we get the bifurcation class right, we should be able to plot the data directly there. So we were very surprised by this. I mean, extremely surprised. And so that's basically the talk. I think I finished kind of on time. And so here, I asked ChatGPT again, can I use Wigner's ideas to describe uranium using random matrices to describe living systems? ChatGPT is not so optimistic about this, but we'll ignore the oracle. We'll be heretics. The heretics are a very misunderstood group in history, I would say. I come from the Silvia Federici School of Historiography. If you haven't read her, she's very interesting. She's trying to reclaim, she's like this very interesting feminist historian who's trying to reclaim all these heretics and all these people who are dissidents. So our modern day oracle doesn't think it's very interesting, but I still think, we'll keep on plugging away. And as I said, that's the group. Everything I do is because of the group. The stuff I showed you on single cell stuff is done by Maria, and Emmy and Sarah are very involved in the microbial ecology stuff. Jason, who's now at a startup run by Josh, was also very involved in many of these things. And Krishna is a new postdoc in the lab working on stuff I didn't talk about. Stuff about differential Gillespie algorithms and using transformers instead to try to think about genotype to phenotype fitness maps. So if you're interested in any of that stuff, and these people gave me money. And thank you for listening. I almost was on time.
That's a blow.
Okay, hey, I really like the second part. So I'm curious, well, not that I didn't.
I love it.
But I guess that one thing that comes to my mind is that, when you describe the kind of Waddington landscape, you just described it as a dynamical system, and then you described it with this modern Hopfield model that has an energy potential. And then in the setting of neuroscience, there are, some neural network dynamics can be described with energy potentials and others can't. And sometimes there's very simple interpretations if you don't have an energy function that are like really convoluted if you try to describe it in terms of an energy function. And so I guess my actual question for you here is I don't think you're trying to be too literal with it. You're just trying to say you're trying to get the bifurcation class. And so I think my question for you is it clear that this class of dynamical systems is rich enough to include all of the various bifurcation types that you would want?
Yes, because we don't actually, I said it's modern Hopfield, but I just want to point out the modern Hopfield is when you don't have these bifurcation things. Then you have a Lipinov exponent. Lipinov. You seem to know something about modern Hopfield networks, yes?
Yeah.
Okay. So if I make this just a square potential, then you have Dimitri's, Dima's, all these energy functionals. As soon as I put the bifurcations in, it can't have a Lipinov because you're basically, of course, killing attractors. So it's a generalization of the Hopfield model. And as long as you put any normal form here in, we have never seen it not work. We do not understand why.
So my question part also is to the left, where you've got the derivative of x is being determined by the derivative of v.
Yeah, but this is fine because this is fine. This is just a continuous version of the discrete update of a normal Hopfield. This is fine. This is in this follow-up paper that Dima wrote where they have the hidden neurons. This is basically the same equation they wrote down.
But I guess going back to the neural network setting, there's a certain neural network, like if you have an asymmetric neural network, but you can't always write that as the derivative of any energy function.
Right. So that's the funny thing, that this softmax kind of is asymmetric, right? It's not like there is a funny trick here, which is that I left it out, which is that if you're technical, we use this funny thing where because the memories are uncorrelated, we make a correlation matrix, we make two things, and the left and right aren't the same. So it's much closer to, it's almost like a transformer, but it's almost like an attention layer, but it's not quite. I don't think these are particularly expressive. I think if they were too expressive, we would have to fit them. They're actually very non-expressive kind of things, but because we don't have to learn the patterns, I specify the patterns. So the expressivity doesn't matter. And the other thing I would say is that we don't know how to we haven't asked about whether we can get chaotic dynamics out of these. So we haven't thought about the bifurcations to chaos, but all the codimension 1 and codimension 2 bifurcations, right, without chaotic dynamics, which is what somehow I can't figure out if it's a result or it's an assumption that David Rand and Eric Sigia and these guys are making, that's what should describe self-fate. Those all work here. But I don't understand about I don't know about the chaotic bifurcations. That's what I would tell you.
Yes.
Or codimension 3.
So maybe can I just say something on that topic to bolster your use of this? So at least what Rand and Sigia tell us, that there's a theorem, Morse-Smale theorem, that says that if you don't have periodic loops in a system, if you don't have periodic orbits in the system, then you can always write it as an almost gradient system. And that's the sort of that's the thing that we're stepping on top of. That's what Pankaj is stepping on top of.
Except there's this nonlinearity in front, so it doesn't.
As I said, almost gradient, like we can,
I don't understand the math of this. It is surprisingly simple and rich at the same time. There's no theorems as far as I can gather.
I guess in a neuroscience setting, you get lots of
periodic.
Yeah.
Totally.
So you don't get that in.
Yeah, you don't get that.
This is very directed.
Very directed. There's not. It doesn't have these kind of chaotic, like Larry Abbott style chaotic things. That is much closer to actually the dynamics of the consumer resource models before. They're almost identical. You can map them to each other. It's kind of interesting.
Can you turn this around? So presumably the cell atlases are sort of final, stable states, cell attractors, whatever you want to call them. But the bifurcation diagram, the bifurcation pictures you're showing seem to indicate these transient states. So can you identify something biological about them?
So this one is kind of interesting because we think this has to do with the fact in the injury repair program in lung, AT2 can turn back into AT1. actually in response to repair. So we think this transient state is something like a canalized state that is there, but you'll never see it. And some of the cells, when you look down at this diagram, you see that most of the highway of these cells, so those cells here are these cells that get stuck. This resolution doesn't have high enough resolution, but they get stuck here. So there's like a super highway of cells that go to AT1 and AT2, and some of them kind of get stuck because there's this leftover thing that we think has to do with injury repair. So you don't really want to go there, but you kind of get stuck there because there's this valley and if there's enough stochasticity. So these are the kind of things that we think you might be able to design experiments with if we plot the single cell RNA-seq that way. And as you point out, this is all based on time series data. So we've colored these cells just by how much AT1 they have because it's hard to see 3D plots if you don't color them by something. So this is just AT1 marker gene expression. That's what the light color says, but it's essentially, yeah. So we think it has to do with the fact that there's a lung repair program underlying it where these cells are present, but even in development, sometimes you get stuck in these things. And then when you get the air-lung interface, which is what happens here, it's like post day 3 and 7, they kind of disappear. So I think there are stories you can tell yourself. How to turn them from stories to real understanding, I don't know, but I must say it's about as excited as I have been about anything in a long time. This stuff.
Okay, let's thank the heretic again.